Hello everyone, welcome to the new session of Role of Genetics in Plant Breeding Part 2. So, in this part, we are going to study about the different methods in plant breeding, right? So, we are having different methods like selection, hybridization, polyploidy, induced mutation, and biotechnological methods. So, these methods we are going to study at an introductory level. We will have a separate session to understand these methods in great detail, right? So here we are going to just know about what are these methods in the plant breeding. So before going to the selection method and the hybridization method, let us understand about the cross-pollination and the self-pollination, as these two things are very, very important to understand the different methods in the plant breeding. So, the pollen is transferred to the female reproductive organs of the seed plant, resulting in the fertilization and the reproduction in a process called pollination. So, what is pollination? The male part of the flower, that is the anther, is having pollen grains, and the transference of the pollen grain to the female reproductive part of the flower, that is the pistil. So when it get transferred to the stigma part of the pistil, then it is called as pollination. This is called as pollination, right? So we are having different types of pollination. The two main category of the pollination is the self-pollination and cross-pollination. Self-pollination and the cross-pollination. This diagram is very clearly shown that the self-pollination and cross-pollination. In the self-pollination, in one individual flower only, that is having both male and female part, the pollen grain is transferred to the stigma of the same flower on the same plant. Then it is called as autogamy. This is called as autogamy, right? And when the pollen grain is transferred to the stigma of another plant, another flower on the same plant, then it is called as geitonogamy. So, self-pollination, this is again a type of self-pollination, right? That is the pollination occurring on the same plant but different flower. Here, the pollination is occurring on the same flower, right? So there is just a difference that in the autogamy, the pollination takes place in the same flower, whereas in the geitonogamy, the pollination takes place on the two different flower on the same plant. That is the geitonogamy. But both are self-pollination. Now, when the pollen grain from one flower of one plant is transferred to the another flower of the same type on the different plant, then it is called as cross-pollination. This is also known as xenogamy. Okay, so there are two different types of pollination, that is cross-pollination and self-pollination. In cross-pollination, the reproduction is called as allogamy, and in self-pollination, it is called as autogamy or geitonogamy, right? So in cross-pollination, pollen is transferred from the plant to plant by an insect or by the wind, both called pollinators. So in some of the plant, we require different agencies like insects or wind, sometimes water also, right? So in different types of plant where they are present, whether they are present in the aquatic environment or whether present on the terrestrial environment, okay? So according to that, they can be pollinated with the help of insects, winds or water agency. From pollen grains of anther to the stigma of the flower of different plant of the same species. Okay, so during self-pollination, plants stamen shed pollen on to its own stigma during the process. So in self-pollination, what happened? The pollen grain which is present on the anther is fallen on the stigma of the same flower. Okay, so the pollen grains are transferred from anther to the stigma of the same flower. This is the self-pollination. For cross-pollination, they are brightly colored petals with nectar and scent. So this is one of the peculiarity, or you can say the characteristic of the flower that are cross-pollinated. So brightly colored petals. The petals are generally brightly colored because they have to attract insects. Same, they are having nectar and scents and long stamen and pistils so that the stamen could be clearly visible to the insects. Same with the pistil, right? So they are brightly colored, having their nectar or scent and their long stamen and pistil. This is the adaptation of the insects pollinated flowers, that they are having the long stamen and the pistils like this. So this is the one of the figure showing the long stamen, right? And sometimes the female part is quite longer than the male part. That is the anther. Okay, so here the anther are longer, and in this case the pistil is longer, right? So self-pollination, it include much smaller flowers. So generally the flower which are not very brightly colored, not having the nectar, because they do not have to attract any insects. So they are quite smaller. And self-pollination may be seen in some legumes such as peanuts, orchids, peas, sunflowers, wheat, oats, potatoes, peaches, and several more. Okay. The two types of pollination can be found in many different plants. Okay, so cross-pollination occur by the insects for the several plants such as apples, grapes, plums, pears, raspberries, strawberries, daffodils, tulips, and other plants. So these are the different plants in which insect is required for the pollination. So the cross-pollination occur by wind for the different plants like grasses, catkins, dandelions, maple tree, and few others. These plants do not have any scent in them. Okay, not brightly colored flowers. So generally they are pollinated with the help of winds. Cross-pollination occur in either perfect or imperfect flower. Generally the flower are imperfect. Means the male flower is different and the female flower is different, right? So that is why they are called as imperfect. But sometimes they are perfect also. That is male and female part present on both on the same flower. Both the reproductive part is present on the same flower. But sometimes they are unisexual flower also. So most flowering plants use cross-pollination. Generally, we are having the cross-pollination flower. So peas, sunflower, and orchids only use self-pollination if cross-pollination is not successful. Generally, they also favor the cross-pollination, but sometimes when there is no cross-pollination, they can undergo the self-pollination as well. Cross-pollination result in more variety of the species. Very, very important. Cross-pollination gives results in the more variety of species, allowing for more diverse appearance of the different plants and flowers. Why we can say that they are having more variety of the species? Because there is a mixing of the chromosomes. They are the mixing of the genetic material, and the new varieties can develop, right? But in the self-pollination, the chromosome is same type. The chromosome is same type, whether it is the chromosome which is present in the pollen grain or the chromosome which is present in the female reproductive part. that is having the same chromosome. So no new varieties can develop, right? But in the case of cross-pollination, always new varieties of the species develop. It favors that the new varieties of the species, formation of the new varieties of the species. The genetic information of plants are combined, but it mostly rely on the pollinators such as insects and the wind moving the pollen from plant to plant. So this is very, very important. So insects. And if we talk about some insect, then we must talk about the bees. Bees are wonderful pollinators. In one of the studies, it came that if the bees are removed from our environment, then our 80% plant will not be pollinated. No pollination in 80% of the plants. So there will be scarcity of food. So now we can understand how these insects are important. Okay, these insects are important because they are very good pollinators. And the very important, bees. In self-pollination, the results allow for more information, uniform progeny, and permits the plants to be less resistant to disease. So here we can see, in addition, since the plants do not need to use energy to attract pollinators, they can spread beyond areas where the pollinators are found. So here we can see the cross-pollination. Okay, so pollen from the stamens sticks to the bees as it visits to the flower. Okay, it bees to the body of the bees, and when it moves to the another flower, that pollen grain accidentally can fall on the stigma, right? So in this way, the pollination takes place. So in cross-pollination, there are very large number, but in self-pollination, there are very small number of pollen grains. Because why they are having the large number of pollen grains in the cross-pollination? Because large number of pollen grains are produced so that it can be transferred to the stigma of another flower. So to have a more probability that larger pollen grain could stick to the body of the pollinators and they can be transferred. And if they are having less number of pollen grain, then their transfers to the another flower will be affected. But why they are having less pollen grain in the case of self-pollination? Because they do not need any agency. So only with the help of some pollen grain also they can have pollination. Right? So let us talk about some methods of the plant breeding. So first of all, the conventional method. The plant breeding is defined as identifying and selecting desirable traits in plants and combining these into one individual plant. So if we are talking about the conventional method, so this was the main thing, that first we should do the identifying the desirable character or the desirable trait in one plant. So I'm just taking one of the examples. Suppose I'm having a wheat plant, right? That is more resistance to the disease. So this is the peculiarity of this particular variety or a species of the wheat plant. Resistance to the diseases. But their grains are smaller. So their grains are not so useful, but this thing, resistance to the disease is important character. So we identified this wheat plant in such a way. Now I am having one more wheat that is having greater size or the larger size seeds, as we can see in the figure like this. Okay, large size grain, but they do not have the resistance to the disease. They are susceptible to the disease. Okay, so what character we want from this one, that is the resistance to the disease, and here large size grains. Now I want a plant that should have both these traits, so we can have a breeding in these two plants, and we can have a new variety of wheat that can have both large grains as well as disease resistance. Okay, so this is the thing. So identifying and selecting desirable traits is very, very important in plants. So here we have identified the resistance to the disease in this particular wheat variety, and in the second, large grain size. So we can combine these two into one individual plant. So Mendel's laws of the genetics provide the scientific basis for the plant breeding. So Mendel experiment, Mendel results, okay, a basis for the plant breeding. So that is important point. All traits of the plants are controlled by the genes located on the chromosomes. So the method of conventional plant breeding is considered as manipulation of combination of the chromosome. So we can have a chromosome here which is having disease resistance, and here we can have a chromosome with gene having larger grains, right? Larger grain size. So with the combination of these two characters in one particular wheat variety, we can have a good variety of wheat that is having both larger grains as well as the disease resistance. So such varieties are desirable for the humankind. There are three procedures to manipulate the plant chromosome combination. First one is the selection. Selection is the most ancient and the basic procedure in the plant breeding. In generally, it involves three different steps. What are these steps? First one, large number of selections are made from the genetically variable original population. So, from the original population, we have to select the large number of the genetically variable plant or the desirable plant. Second, progeny rows are grown from the individual plant and selections for the observational purposes. So we could have the progeny for all the selected varieties, and we can select the observational, and we can select our desirable plant. After obvious elimination, means we can eliminate the one which we do not desire, okay, and we can select the one which is having the desirable characteristics. So the selections are grown over several years to permit observations of performance under the different environmental conditions for making further elimination. So this is a time-taking process in which you have to grow these crops a number of times. You have to record the environmental condition and then select or eliminate the undesirable one and select the desirable one. So finally, the selected and inbreed lines are compared to the existing commercial varieties in their yielding performance and other aspects of the agronomic importance. So these selected lines are then undergo for the inbreeding process. Number of times we will inbreed and we can have a good variety or the desirable variety of that particular species. Next one is the hybridization. The most frequently employed plant inbreeding technique is the hybridization. The aim of hybridization is to bring together desired traits found in the different plant lines into one plant line via cross pollination. So I have just shown you or discussed that wheat plant that is having the disease resistance and the one which is having the larger grain size, that is that can undergo the hybridization technique. So here we have taken one of the one more example, that is the teoscinite parent and maize plant, two different species. Now the cross teoscinite and the maize parent to produce hybrid offspring. Okay, so this is one of the hybridization technique. Okay, so in hybridization there are different steps. So the first step is to generate homozygous inbreed line. This is normally done by using the self-pollinating plants, where the pollen from the male flowers pollinators pollinates the female flowers from the same plant. So every time you have to pollinate, that is the self-pollinating. Okay, and you can have a homozygous inbreed lines, pure form of that plant. Once the pure line is generated, it is outcrossed, that is combined with another inbreed line. Then resulting the progeny is selected for the combination of desired traits. So once the pure line is generated, then you can outcross. You can outcross with the different variety or the different species even. If a trait from the wild relative of a crop species, resistance against disease, is to be brought into the genome of a crop, a large quantity of undesired traits like low yield, bad taste, low nutritional value are transferred to the crop as well. So the result cannot be always beneficial. Sometimes what happens, we are desiring this resistance against the disease. But along with this disease resistance trait, some other undesired trait can also come to the plant, that is low yield, bad taste, or the low nutritional value. So these unfavorable traits must be removed by time consuming back crossing. Okay, that is repeated crossing with the crop parent. So you can do back crossing. Back crossing means you can have a hybrid of that plant and you can cross with the parent, one of the parent, that is the back cross. There are two types of hybrid plant, interspecific and intergenic generic hybrids. So beyond this biological boundary, hybridization cannot be accomplished due to the sexual incompatibility, which limits the possibilities of introducing desired traits into the plants. Now this is very, very important term, that is the sexual incompatibility. Sexual incompatibility means plants do not allow certain pollen grains to get on it. If they germinate also on the stigma, they do not allow to go to the style region. And if they go to the style region, then they do not allow to get enter in the embryo sac sometime. Okay. So this is called the self-incompatibility, right? So beyond this biological boundary, hybridization cannot be accomplished due to the sexual incompatibility. But this is not for all the species. So we can have the interspecific and intergeneric hybrid depending upon how the plants allow the pollination and finally the fertilization. Heterosis is an effect which achieved by the crossing highly inbreed lines of the crop plants. Inbreeding of most crop leads to the strong reduction of the vigor and size in the first generation. So sometimes inbreeding in most of the crops reduces the vigor and size of the first generation. After 6 or 7 generation, no further reduction in the vigor or size is found. So you have to get the number of inbreeding cross to get the greater vigor and size of these seeds. When such highly inbreed plants are crossed with other inbreed varieties, very vigorous, large-sized and large fruited plants may result. So this is very important, that for the number of generations, you have to inbreed that plants. The most notable and the successful hybrid plants ever produced is the hybrid maize. So here we are having a different steps to remember. Selection of the parent plant with the different qualities, removal of stamens from the female flower. So no more contamination of the other pollen grain. Okay, so the collection of pollen grain from the male flower, that is also selected. Okay, and here when you remove this anther of this flower, okay, you remove this anther, then what happened? This plant is just a female flower. And the one which you have selected, the male flower, you take the pollen grain and have dusted on this stigma region, so that you get ensured that the pollination is done by this male flower only. Okay, so pollinating the female flower with the collection of pollen grain covers the mother plant with the polythene bag, and later this particular plant is covered, or the flower is covered with the help of polythene bag, so that no contamination, no other pollen grain could come and pollinate that flower. So in the next step, cover the mother plant with the polythene bag. Collection of mature seeds from the mother plant. So we will collect the mature seeds after the fertilization. Okay, select and grows better varieties of the offspring. So this is the technique of hybridization. Next, polyploidy. Most plants are diploid. Plants with 3 or more complete sets of chromosomes are common and are referred to as polyploids. So the increase of chromosome sets per cell can be artificially induced by applying the chemical colchicine. So this is very, very important. You can increase the ploidy of a particular plant by applying this chemical colchicine, which leads to doubling the chromosome number. So if the plant is having XX, YY, okay, suppose, then it can have XX, XX, YY, YY. So the chromosome got doubled. Generally, the main effect of the polyploidy is increased in the size and genetic variability. So polyploidy leads to increase the size and genetic variability of the plant. So on the other hand, polyploid plants often have a lower fertility and grow more slowly. So this is the drawback. That they are having lower fertility and grow slowly as compared to the diploid one. Some of the examples are strawberries, wheat, and mango. So in these plants, polyploidy has been generated. Induced mutation. This is another method of the plant breeding. Instead of relying only on the introduction of the genetic variability from the wild species gene pool or from other cultivars, an alternative is the introduction of mutation induced by the chemicals or radiation. So here in the ancestral crop, this is showing the mutation, causing mutation in this particular part of the DNA. Now this is the mutated DNA and this is the modern crop. Now you can see here the seeds are quite smaller and the plant is larger. But here the plant is smaller and the seeds are larger. So due to this mutation in the DNA, these changes can occur. So this is called induced mutation. We are causing this mutation in the plant with the help of some radiations or chemicals. The mutations obtained are tested and further selected for the desired traits. So sometimes what happens, these new plants after the mutation, they are tested whether they are having the required traits or not. If they are having that required trait, they are selected and having the desired traits, and the new species or the new variety of the crops is introduced. These site of mutation cannot be controlled when the chemicals or radiations are used as an agents of the mutagenesis, because the great majority of the mutations carry undesirable traits. So again, there is not always you will have the desirable traits, some undesirable traits, because this mutation, whether you are taking the chemicals or the radiation, sometimes they changes some of the genes that are very important for the plants. So that is undesirable traits. So this method has not been widely used in the breeding programs. Now comes the biotechnological method. Finally, this method is a discipline which deals with use of living organism or their products. So in this particular, a desirable set of genes is introduced in the host organism. Okay, so in this wide sense, also traditional agriculture may be seen as a form of biotechnology. So this particular branch of science is nowadays taking a very great step in the agriculture field also. So the European Federation of the Biotechnology defines biotechnology as the integrated use of biochemistry, microbiology, and engineering sciences in order to achieve technological application of the capability of microorganism, culture tissue cells, and the part of. So here we are having different integrated technology, that is the biochemistry, microbiology, and engineering science, and this comes as a new technology that is called as a biotechnology. So in recent years, biotechnology has developed rapidly as a practical means for accelerating success in the plant breeding and improving the economically important crops. So nowadays, this technology is widely used in the breeding program. Okay. So this is all about this plant breeding. Okay, we will have one more session to know the details of the selection process, hybridization process, induced mutation, and the biotechnological method. Okay, till then, thank you very much.